Good day to you all. You're most welcome to today's topic 3.1, which is on leukocytes. I'm Dr. Penina Nsamba, a lecturer in the Department of Biomedical Lab Technology and Molecular Biology, abbreviated as BLT, in the School of Biosecurity, Biotechnical and Laboratory Sciences, SBLS, in the College of Veterinary Medicine, Animal Resources and Biosecurity, abbreviated as COVAB, in Makerere University. And this is a photograph of me. Most welcome to today's topic, 3.1 leukocytes. And we shall start with the morphology and function of the leukocytes. The contents will include an overview and justification for their study. Then we shall look at granulocytes, morphology and function, agranulocytes, morphology and function. The learning objectives. By the end of this lesson, you'll be able to describe the morphology of the different leukocytes and explain the function of each. Let's start with an overview of the leukocytes. They are white blood cells and they can be classified according to size, nucleus shape, or the cytoplasm color and existence of granules therein. The different types of leukocytes in this lesson will be classified based on the granules, and this will result in the two following broad categories: the granulocytes and the agranulocytes. The granulocytes are the neutrophils, eosinophils, and basophils. They have granules in the cytoplasms and they'll have a segmented nucleus. The agranulocytes are the lymphocytes and monocytes. They have no granules in the cytoplasm and the nucleus is entire. It's not segmented or lobulated. Let's look at the first granulocyte. And as a reminder, they have got cytoplasmic granules. The neutrophil. The picture above shows a three-dimensional view of the neutrophil, and the one below shows what you'd see in a thin blood smear. The description of the neutrophil. They're segmented. It has a segmented nucleus, or we can say it has a lobed nucleus, typically with 3 to 5 lobes, as we see in this thin blood smear. They have got dust pink granules in the cytoplasm. An increase in the number of neutrophils in the bloodstream is called a neutrophilia, while a decrease in the number of neutrophils circulating in the bloodstream is called a neutropenia. The functions: the neutrophils respond to bacterial infection and inflammation, among others. The immature stages of the neutrophils are called band cells. The nucleus is either S-shaped or C-shaped. The photograph on the left shows a C-shaped nucleus for a band neutrophil, while the one on the right shows an S-shape for the band neutrophil. In these immature stages, it's possible to see the granules. Let's reflect back on what we've just studied by looking at the following study question. A. Describe the granules found in a neutrophil cell. Hint will be the color and size. B. Describe the nucleus of a mature neutrophil cell. Hint would be in the shape. C. Describe the nucleus of a band cell. Hint would be the shape. The answers: the granules in the neutrophil are dust pink. The shape of a mature neutrophil nucleus is segmented with 3 to 5 lobes. The nucleus of a band neutrophil is either C-shaped or S-shaped. I believe you got the answers right. If you have any problems, reflect back on the previous contents of this video. Let's look at the second granulocyte. It's the eosinophil. The picture above shows an eosinophil in 3 dimensions. The one below shows what you'd see in a thin blood smear. So typically the eosinophil cell has a segmented nucleus or a lobed nucleus, and it consists usually of 2 lobes. It has large reddish orange granules in the cytoplasm. They're clearly seen in both pictures. An increase in the number of eosinophils circulating in the blood is eosinophilia. A decrease in the number of eosinophils circulating in the blood is an eosinopenia. Functions: they respond to parasitic infection or allergic inflammation, among others. Let's reflect on what we've just studied about the eosinophil. A. Describe the granules found in an eosinophil cell. Hint would be the color and size. List the function of the eosinophil cell. C. What disease syndrome can lead to an eosinophilia or an increase in numbers of eosinophils circulating in the bloodstream? The answer to part A: the granules in an eosinophil are large and bright orange or red. Part B: functions of an eosinophil cell is to respond to parasitic infections. Part C: intestinal worms is a syndrome where we possibly may see an eosinophilia. Still study questions. Here we want you to compare the eosinophil and the neutrophil cells. A hint would be to consider the nature of the granules and the nucleus lobes. Typically, this is a take-home exercise, so you'll do it in your revision groups or your discussion groups and submit it as an assignment. Let's look at the third category of granulocyte cells. These are the basophils. Here we've got a picture of what you'd see in a thin blood smear under a microscope, showing a basophil. Typically, it is segmented. It has a segmented nucleus of 2 lobes, and we see large bluish or purple granules in the cytoplasm, and these partially obliterate the nucleus and cytoplasm. Increase in numbers of circulating basophils in the bloodstream is called a basophilia. The functions of the basophils are to release histamines and heparin during allergy reactions. So that ends the granulocytes. We're going to move on to the agranulocytes. The first category we're going to look at are the lymphocytes. As a reminder, they do not have cytoplasmic granules. The pictures here show lymphocytes in a thin blood smear. The one on the top shows a normal lymphocyte. The one below shows an activated lymphocyte. The nucleus is round, it's entire, and it fills most of the cell. So we've got a high nucleus to cytoplasm ratio. The cytoplasm is blue colored. Now, if you look at the activated lymphocyte at the bottom, the nucleus has been pushed towards the side, revealing more cytoplasm. The activated nucleus is active and ready to respond. The functions are to produce antibodies, B cells. Increases in lymphocytes circulating in the blood is called a lymphocytosis. Decrease in numbers of lymphocytes circulating in the blood is called a lymphopenia. The second category of a granulocyte is the monocyte. It is large in size compared to the other white blood cells. The picture above shows a three-dimensional view of a monocyte, while the one below shows what you'd see in a thin blood smear. And if we look at the picture at the bottom and we compare the size of the monocyte to that of the red blood cells, we find that we could easily fit about 3 to 4 red blood cells in the space of this monocyte. That's why we are describing it as large compared to the other white blood cells. If we just go back a minute and look at the lymphocyte actually may be equivalent to the size of a single red blood cell when it's normal and non-reactive. The reactive lymphocyte, on the other hand, would be actually equivalent to the space of about 2 red blood cells. If we look at the monocyte, using the photograph below, we can easily say that it actually takes up the space of 3 to 4 red blood cells. So that's about the size. The cytoplasm of the monocyte is bluish gray in color, and it has got vacuoles. The nucleus is entire, it's not segmented, and it is indented. Sometimes we can refer to this shape as a horseshoe shape. Increase in numbers of monocytes is called a monocytosis. The functions of the monocyte are phagocytosis and to replenish dendritic and the macrophage cells. Let's review what we've just studied on the monocytes and the lymphocytes. Would like you to compare the morphology of the lymphocyte to that of the monocyte. As a hint, look at the nucleus shape and the unique features of the cytoplasm. Typically, this is a take-home exercise, so you're going to do it in your various study groups. In summary, in this section 3.11 of the lesson, we have considered the heterogeneous nature of the leukocytes. We have described the unique morphology of each leukocyte and highlighted their significance in relation to the function. The next steps: refer to the study portal for the lecture notes, the e-activities therein, and the chapters in the textbooks that we have recommended to you for further reading. This is our source of information. Thank you.